Hello there, Year 9. We've spent a lot of time now in this topic, B4, looking at photosynthesis, the reaction that is a chemical reaction taking place in the chloroplasts of those palisade cells found in the leaf. And we've looked at all of those different limiting factors and how they affect the rate of photosynthesis. And now what we're doing is we're taking that a step further. We're saying, okay, the plant produces glucose, that's its food, as a result of photosynthesis. Now, what does the plant do with that glucose? And there are 5 uses of glucose in the plant. And it doesn't sound necessarily that exciting, but it's a massive favourite question with the exam board. They love telling you one of the uses of the glucose in the plant, and then for a 4 marker, getting you to come up with the other 4. And it was only a couple of years ago when I was writing out the 5 different things that plants do with that glucose that I accidentally spelt out the word scarf. So it doesn't mean anything to anyone but us, it doesn't mean anything to the exam board, but it helps us to remember those 5 uses of glucose in the plant. So as I said, it's scarf, so we've got the S there for some of that glucose is stored as starch. We've got some of that glucose is used to produce cellulose, and this strengthens the cell wall. Some of it is used to build amino acids, and amino acids then build up to make proteins, and the proteins are used for growth. Some of it is used for respiration, which we've talked about. It's the reverse of photosynthesis. We'll be talking about respiration a lot more in the second part of B4. And we've got to remember that all living things respire. So even though we tend to think of plants as just photosynthesizing, they're also respiring at the same time. And then our F is that they are stored as fats and oils, okay? So this is kind of our summary for the next 2 lessons. We're going to be looking at all of those uses of glucose by the plant, and our summary is that we've got this phrase scarf, okay? Starch, cellulose, amino acids, respiration, fats and oils. Now you can write this page out if you want to, just as your kind of summary of the beginning part of this block of 2 lessons, but we're going to look at each of these in turn. And today we're going to look at respiration, and we're also going to look at storage as starch, okay? And we'll cover the other 3 next lesson. That's just another piece of information there, that to make proteins, plants also take in other minerals, for example, nitrate ions from the soil, okay? But again, they're not referred to as food. The food for the plant is the glucose, which they make themselves using that process, photosynthesis. So if you need extra time on this slide, then pause now. Okay, so we're going to look at respiration first of all. It's the reverse of photosynthesis. So like I said, we spent a lot of time looking at the photosynthesis equation, the word and symbol equation, and if you turn that equation round, then you have respiration. Okay, so taking that glucose, some of the glucose that was made in photosynthesis, taking some of that oxygen, and then releasing the energy from the glucose, and the byproducts are carbon dioxide and water. And we said so that we don't spend a long time balancing that equation, we just remember that all of the small molecules have that number 6 put in front of them. We will need to memorise that chemical formula for glucose, C6H12O6. But we are going to revisit respiration in more detail later on in the topic. So for now, I'm just going to leave it there. If you want to write some notes from that page, that would be really useful, even if it's just the word equation, just acknowledging that one of those uses of glucose is respiration. And we're just talking about the plant right now. If you need extra time on that slide, then pause now. Okay, so that was kind of our thinking activity, although we did do some writing. Our title for the day is How Do Plants Use Glucose? Don't forget to write the date as well. So I've already started to explain that plants do photosynthesis. And the purpose of this photosynthesis is to make glucose. And there are 5 things, purposes, functions that the plant uses that glucose for. We're looking at 2 of them today: respiration and starch. Hopefully you've got your title written down. If you need any extra time, then pause now. Okay, so we've got a bit of graph interpretation here. This is a plant, and we can see that we've got this constant, unchanging rate of respiration. Okay, the red line, it stays constant, so it doesn't matter if it's night or day, plants respire at a steady rate. And we can see that across the day, photosynthesis increases and then it decreases. Okay, now that increase in photosynthesis is going to relate to sunlight. We know that plants need to take in that sunlight in order for photosynthesis to occur. The light is absorbed by the chlorophyll in the chloroplasts. So when we are at dawn and dusk, we see photosynthesis dropping off. So the reason why I think in the past we tend to say all plants do photosynthesis and we forget about the respiration is because most of the time the rate of photosynthesis far exceeds, okay, that means there's a lot more of it than there is the rate of respiration. So by comparison, you can see most of the time photosynthesis is a much more prominent, much more obvious, if you like, reaction that's occurring rather than the respiration. Okay, so we've been asked here to compare the rates of photosynthesis and respiration over the day. I've done a lot of talking there. What I'd like you to do, please, is to draw a sketch graph and then to maybe just summarise with 3 short sentences what the graph is showing us. So we had respiration remaining constant, we had photosynthesis only occurring in daylight hours, and we had the more light, the higher the rate of photosynthesis, as we can see by the curve on the graph there. Okay, you're probably going to need some extra time there, so if you need extra time, then pause now. Okay, now this is in a different form of basically the same information we've just had. Now what we've got to picture is that we're looking at plants in an enclosed environment. Okay, what I mean by that is we're looking at them in a greenhouse. Now if we could monitor the concentration of oxygen and carbon dioxide in that greenhouse, how would we see those 2 different gases fluctuate, change over night and day? Now, we kind of have to refer to our previous graph to help us with this. So we said that in the daytime, there's lots of sunlight, so there is more photosynthesis. Now, photosynthesis produces oxygen. So we can see that oxygen levels in the daytime increase. That's because photosynthesis is happening at a higher rate. We also know that at night, no, actually I want to continue to talk about daytime. Sorry. So we also know that photosynthesis uses up carbon dioxide. So at the same time as the oxygen increasing by that increased rate of photosynthesis, we can see the carbon dioxide being used up and dropping. Okay. Now, we'll go back to this graph and we can see that at night time, the photosynthesis rate completely drops down, so we are more aware of the respiration. Respiration hasn't changed, but relatively speaking, it is at a higher rate than photosynthesis. So relatively means by comparison. So by comparison to the rate of photosynthesis, at this point here, there is a higher rate of respiration. Okay, so now let's have a look at that second part. Okay, so at night time, there is a relatively higher rate of respiration, and respiration releases carbon dioxide, so we see that slight increase in carbon dioxide at night. Respiration uses up oxygen, so we see that decline, the decrease in oxygen at night. So the reason why this question here is a 4 marker is because we've effectively got 4 separate parts of the graph that we can talk about. Okay, so same as last time, we're going to sketch the graph, try and use some different colours, try and use some different colours, and then try and write 4 bullet points to describe the 4 parts of the graph. If you need extra time, pause now. Okay, that's us done talking about respiration for now. Like I said, respiration comes up later in the topic as well. Right now we're going to talk about starch. Now, glucose is a small molecule. Okay, we can see it by this picture here. So these are glucose molecules. They're very small, they're soluble. When we get into B2 at the beginning of year 10, we'll talk about how they are able to, because they're small and soluble, move across from the small intestine into the blood. And plants store those glucose molecules all stuck together in a row called starch. And here is our starch molecule. It's a large molecule and it's insoluble. Okay, they can't store it as sugar, as the glucose, they have to store it as these long chains called starch. So some of that excess glucose is stored as starch. Starch, I always say, starch can be used when the plant cannot photosynthesize. It's kind of like a midnight feast. So when the plant is photosynthesizing in the day, it's actually making more food than it needs. So some of that glucose is being used straight away on these 5 processes, and some of it is being stored for later as this molecule called starch. Now humans do this too. We don't store it as starch though, we store it as a substance called glycogen, and we'll talk about that one later in year 10. So let's get some notes from this page then, please. And if you need extra time, then pause now. Okay, so this is a practical that we would do in the classroom. It's a shame that we haven't been able to do it. I haven't got a demonstration of it, but I have got some diagrams, and we will have a go at doing this when we get back to school. So the big question for this investigation is, what differences will there be between a plant stored in the light and a plant stored in the dark? Now, although this is not a required practical, it's another favourite with the exam board. They really like questions talking about the storage of starch. So what we do is we take 2 plants, same species of plant, not that it really matters, and we store one in the dark for 24 hours, so we keep it in a cupboard covered over with a bin bag, and then we store one under a lamp for 24 hours. We tear off a piece of each of the leaves from each of the plants, and we investigate for starch. Okay, so if we write down our big question, now we could take a moment here to hypothesize, that means to predict with a reason, whether we would find starch in both plants, just the plant that's been in the light, or just the plant that's been in the dark. So thinking about what we already know, where would we expect to find starch? In the plant in the light, in the plant in the dark, in both, or in neither? Have a go at writing down what you think you might find. If you need any extra time, pause now. So we've got these things here called variegated leaves. And variegated leaves have white or yellow patches around the edge. You might see them on something like a geranium or a spider plant. We've talked about these before. This isn't a great picture of a variegated leaf because you don't often get the patches like that. It's usually around the outside. But if there is a variegated leaf where there is no chlorophyll in parts of the leaf, those parts of the leaf cannot photosynthesize. Therefore, you will not find any starch in those parts of the leaf. So here, here and here, we can see when it's tested with iodine solution, which is the chemical test for starch, it remains orange. Okay, so iodine solution is an orange chemical, and when it is in the presence of starch, it will turn blue-black, as we can see in these regions. When there is no starch, it will remain orange. So you can see on those variegated parts of the leaf where it is there's no chlorophyll, we can see there is no starch by the fact that the iodine solution has not changed colour. Now that's really important moving forward in this investigation, because we need to know what our chemical is, iodine solution, and what are the positive and negative results. Okay, so the fact that it goes blue-black with starch and the fact that it stays orange without starch. Okay, if you need extra time on that slide, pause now. So here's our method. This is what I would put up on the board as we do our investigation. So we'd get our Bunsen burners out and we would boil some water. We'd have our two sections of leaf. Don't usually take the whole leaf, otherwise we end up destroying a plant in one lesson. So we just take part of a leaf and we put that into the boiling water. And the point of that is it breaks down the cell wall. It allows the chemicals to enter the cells of the leaf. Okay, so we leave that to boil for 5 minutes in the water. Now, the next really important thing to do is to get those Bunsen burners turned off, because we're going to add into a boiling tube some ethanol. And ethanol is highly flammable, so we wouldn't want any spillages and we wouldn't want those Bunsen burners on, because that could be quite hazardous. Okay, another really important question that the exam board like to talk about. They like to talk about why ethanol is dangerous. Okay, it's because it's flammable. So we've now put the leaves into a boiling tube, and into that boiling tube is ethanol, and that's gone back into the hot water bath of the beaker. And we actually, when I did this with the previous group, we actually found that the ethanol started bubbling away. It was actually boiling, even though the water wasn't boiling, because it has a lower boiling point than water. And we leave it there for about 5 minutes or so, and it allows the chlorophyll to come out of those cells. Okay, why is that important? Well, it's important so that we can see the colour that the iodine solution changes. So we've got hot water to break down the cell wall, and then we've got the ethanol to allow the chlorophyll to escape, which then allows us to see exactly the colour change of the iodine solution to discover, is there starch in this leaf or not? Okay, if you need any extra time on this slide, pause now. Okay, so we've got parts of the experiment and the purpose of this part of the experiment. Now, this bit we'll take it as an optional activity. Okay, so you don't have to write notes on this if you're happy with this information. So, boiling the leaf in water was to break down the cell wall. Turning the Bunsen burner off was because ethanol is very flammable. Leaving the leaf in ethanol removes the chlorophyll from the leaf, and adding iodine to the leaf lets us see if the leaf contains starch. So that was the kind of reminder activity of why we did each stage of that experiment. Like I said, optional. If you want to add those notes to your book, you can, but I think we may well have covered those already with the method from the previous slide. If you need extra time, then pause now. Okay, so we've done our experiment in theory, and these are our two leaves. Okay, so the plant leaf on the left, the iodine solution has remained orange. This was the plant that was in the dark. Okay, so it stayed orange. That means there's no starch. It used it up on its midnight feast. The plant leaf on the right, the iodine solution has turned blue-black. This means there's plenty of starch in that leaf, and this is the leaf that was in the light. Okay, it's been photosynthesizing lots. It's stored loads of that glucose as starch, and we can see it there by the changing colour blue-black. Now this slide is really important. Normally I would take a photograph of your leaves and then print it for you and put it into your book, but as we haven't had a chance to do it, I think this is really good as our results page to put into our books. So make sure you please draw 2 simple leaves, and we could also annotate on here that the leaf on this side, the left side, this is the one that was in the dark, and this one over here that's full of starch, this one was the one that was in the light. I should have added that to my slide. If you need extra time, then pause now. Okay, we've got a 4-mark question here. I do appreciate this lesson is quite long. I've tried to cram lots in. I didn't realise it was going to be quite this long, but well done for persevering with it. Maybe have a little comfort break and then come back to this last part of the lesson. So this is exactly the same principle as what we did, except rather than using 2 different leaves, they've covered up the leaf with a piece of black paper and clipped it on there, and it does exactly the same job. And what we see is that part where it's been covered up by the black strip of paper, all of the starch that was there was used as that midnight feast. Now, I keep saying midnight feast, we can't write that in the exam. We need to say that that area wasn't photosynthesizing, wasn't producing glucose, and therefore didn't have that starch. It used up the starch that it had. So we can see there's that area there where the paper had been, and the iodine solution has not changed colour because there is no starch present. However, interestingly enough, there is starch either side. I could definitely understand seeing starch in this area here, but it's really interesting that this area's got starch as well. It's been photosynthesizing because it's received that light. Those cells have done it, even though it's right at the end of the leaf, at the end of the plant there, it's still photosynthesized and we've still got the starch, okay? This one, just 4 quick bullet points. I know we're writing a lot today, but 4 quick bullet points to just show that you understand that this occurs in a leaf in different we've kind of had the variegated leaf, we've had the different plants, one in the light, one in the dark, and now we've had one where we've taken the same plant and just put it in the light but then made part of it dark, okay? So that's what that's showing. If you need extra time, then pause now. Okay, now this is our big conclusion, okay? And this is our final bit of today's lesson. What we need to do is imagine that we've done all of that lovely practical. We've had a go, we've had the Bunsen burners out, and we've had the leaves in there, and we've tested them. We're now getting all of our ideas together. And the way that it'd be nice to do this is to say, okay, we're going to write about between half a page and a page in our book, and we want this to be a nice kind of continuous paragraph of writing, maybe a couple of paragraphs, using these questions to help us. So don't write 1, 2 and 3, do some nice full sentence flowing paragraph to prove your understanding of this practical. And I would like you to take a photograph of this page and send it to me on Show My Homework, please. So when you've done this write-up, which is going to be between half a page and a page of writing, answering these 3 questions, you're going to take a photograph, upload that onto Show My Homework so I can see your work. So we had that question, we've written this question already. What differences will there be between a plant stored in the light and a plant stored in the dark? Okay, and first question, or first part of this, is explain how starch is formed in leaves and refer to photosynthesis. Okay, so in that section we're going to be talking about photosynthesis, what that produces and how that becomes starch. Okay, I'm giving some information but not all of it because this is just recapping what we've already done today. We're then going to identify what the iodine solution test shows us. So in that we're going to identify the colours and what that tells us about whether there's starch or no starch, and that's for both colour, well, the original colour of the iodine solution and the colour change. Okay, again, full sentence answers. And then the last bit, describe the results of each plant and explain what this means. So what did we see from the leaf that was in the light? What did we see from the leaf that was in the dark? What colour did the iodine go? And what does that tell us about the amount of starch and where that particular plant was stored? Okay, so we're bringing, this conclusion is going to give me a really good idea of how well you've understood what we've done today. Okay, so that's why I'm not giving too much away. The lesson allows you to be able to answer this, and this is the bit that you're going to submit to me so I can see your understanding. If you're not taught by me at the moment, you're taught by Sir, then please submit this to Sir so that he can read your work, please. Okay, if you need extra time, which you probably will, then pause now. Okay, so that's the end of this lesson for today. I know it was a long one, but quite interesting to see that bit, kind of the step further really. What does the plant do with that glucose? We've learnt about scarf and we've explored two of those, starch and respiration. Next lesson we'll look at the cellulose, the amino acids and the fats and oils. Okay, have a good day. Thanks for listening, and don't forget to submit that work to me.